So hello everyone, thanks for coming along to our very first of the ACCS Knowledge Share Series events for the year 2022. This is actually event number 11 in our series. So we had a lot of success last year with it, and I'm really happy to see you all coming back again this year for some more. And I just a little bit of housekeeping is that, as you know, these events are quite casual. If you want to ask a question, please do whenever you feel like it, put it in the chat or unmute yourselves and just ask it. If you would like to turn your camera off and eat your lunch, please do. These events are recorded and will be added to the OSF repository. So bear that in mind and mute yourself or turn your camera off if you would prefer. That's fine by us. And I'm not going to go on too much about it because, as I said in the email, I was really super excited to have Hoang and James and Nick come along today to talk about this. I have seen just a very brief demo of the IPP image processing portal, and it looks amazing. And so I am very excited to have them take up the full hour and show us all that they have to show about it. And without me going on anymore, I will hand over to them to run today's session. Thanks, guys.
Thanks, Catherine. Hi, everyone. I'm James Springfield, manager of the IMB Light Microscopy core here at UQ. And together with Nicholas Condon and Hoang Nguyen, we're going to be introducing the image processing portal that we've developed. Originally, the portal was designed to help our users, who are mainly wet lab biologists, connect to HPC and large-scale compute in a user-friendly way. But first, I'd like to give you a quick background of why we developed the portal in the first place. So traditionally, the typical workflow for our users from scope to paper involves at least 3 major steps: acquisition, which includes acquiring the data at the microscope, and then transferring the data to secure storage for later retrieval. Processing, where we prepare the data for subsequent downstream use, and this processing often involves multiple repeated steps. And finally, visualization, segmentation, and analysis, where we turn the processed images into something quantifiable for publication. So up until fairly recently, we relied on the vendor supplied PCs, which generally had slow local storage drives and network transfer to both capture, transfer, and store the relatively small data sets we created, which were acquired on PMT and CCD-based sensors. Now, the data was then moved from these acquisition systems to storage and then to desktop class analysis PCs for processing, visualization, analysis. The software running on those PCs was generally running on Windows and not typically written for GPU acceleration, and a lot of the processing took overnight or longer to run. So most of these image processing steps that we were performing were actually doubling the data size, and we were doing it multiple times. So we'd also run into issues with constantly moving ever increasing amounts of data back and forth between storage, analysis, and users' laptops. So all of this was fine up until around 2009 or so. Most of our systems were generating at really low data bandwidths. However, around this time, the first CMOS cameras started coming out, and we started using those on some of our wide field microscopes. Because these cameras were much more sensitive, they enabled a reduction in exposure times and resulted in much higher frame rates. Coupling this with higher pixel counts on the sensor and the possibility of multiple camera systems, suddenly we had access to microscopes that could generate 80 times more data per second. Now, given that IMB microscopy supports a large and diverse user base, and since most of our microscopes are quite niche and they only cater to certain requirements, that required us to purchase multiple systems over a short period of time. And each one is capable of generating vast amounts of data. So our data rate for the facility increased massively. A quick survey across Australia, just looking at facility websites, shows that the total number of these CMOS-based cameras that we know about has increased greatly over the last few years, and most places will actually have more than one of these on their microscopes. 2019 paper by Wojciech indicates light sheets, which are a subset of wide field microscopy, which uses CMOS cameras, is a growing instrument of choice. And the thing with these microscopes is they generally produce data in the terabyte range with an estimated 3 terabytes per week generated. These large data sets often need multiple pre-processing steps before you can visualize and analyze the data. And such big data requires an investment in informatics resources. Many respondents to this survey claim their current systems did not meet their processing requirements. And so we have extrapolated that clearly we've got a nationwide issue that needs to be addressed. So within a short space of around 10 years, our total facility data generation has increased by a factor of 50. Many of our systems routinely produce 1.5 terabyte data sets, and they require subsequent processing, often with multiple steps. Each step often doubles the data size, and since new camera technology is constantly being developed, we know that the problem wasn't going to go away, and that this is also a nationwide problem. So it was time to rethink both our hardware and workflow strategy to make it scalable for the future. To do this, we chipped away at the problem by approaching it from different angles, including acquisition PC hard drives were beefed up to higher I/O NVMe RAID drives, and we also installed faster network cards. We installed fast backplane switches in our building to allow us to move data onto our new local GPFS cache, which also then automatically populates the other caches on campus to allow us to move the data to different resources. We invested in GPU-based HPC to handle processing over big data and began testing rudimentary command line image processing workflows. And we moved to remote computing on HPC-grade servers coupled with open source and commercial software packages to visualize and analyze the data. So we figured we'd given ourselves some breathing space, and we were pretty chuffed with all the hard work we'd done. And then along came Nick, who I'll pass over to so he can tell us his user story and the grief that he brought me.
Thanks, James. Very kind introduction. So my name is Nicholas Condon, and I completed my PhD in cell biology here at the IMB. And during my PhD project, I used lots of different microscopes to monitor changes in membrane dynamics of immune cells when they encountered pathogens. During this time, a younger and probably happier me had the opportunity to travel to Genelia over in the US and use a novel microscope that had just been published. So this was back in 2015, and it was called the Lattice Light-sheet. I got to use the first prototype instrument, and you can see from the pictures on the right here that it really was a prototype instrument at this time with just broken pieces of pipe put around the system to kind of shield the light path the best it can. Even though this was a prototype instrument, we spent just under 2 weeks there and had almost 35 terabytes of data to process. So dealing with that much data back in 2015 was pretty hard. We shipped the data back on portable hard drives, and we then ingested them into this new server that we had with IT called Objective. And that was connected with a 1 gigabit per second network back to those, Mac Pro workstations that we were dealing with. It took 3 years to kind of process and analyze that data to publication. And we did publish the work, and I think it's important to remind ourselves, especially someone like me who's moved from research over to the professional side, that what we do and how we help researchers is actually important, and it can result in important discoveries. Now, I'm a bit shameless self-promoting here, but what we discovered with this microscope is a new method for how cells drink and sample their environment. And that's important for cancer and for immunology. But this discovery wouldn't have been possible without both the invention of the lattice light sheet microscope technology, but also just the ability to process it. And even if that took 3 years to process, speeding up this process will speed up discovery. Okay, so a few people in this audience have probably heard of a lattice light sheet microscope. They may have seen one. They may not really know what we're talking about here. So just to give people a 30-second overview, lattice light sheet and light sheet microscopes tend to illuminate the sample in a slightly different way to a conventional system. Here you can see in this movie a red cell and the green representing the light sheet illumination cycling through. When it's a lattice, that sheet is simply an optical lattice that's moved through. This means that it's gentler on the samples, which means we can image for longer and we can capture data really fast. So we get this, 1 volume a second is pretty standard. Now, because of this kind of weird angle and it's not conventional to normal systems, it requires a lot of computing with these samples. So we have to post-process them through a step called de-skewing, and the data sets really benefit from a process called deconvolution. And I'm going to explain those in a little bit, but here's some examples of the data that can be produced. And this is a cancer cell. Over a long period of time, we're watching it sample its environment. We can drug it and look at different ways it responds, or we can see really highly temporal events like this immune cell reaching up and grabbing that pathogen and pulling it back in to kill it and clear the infection. The data sets kind of are very large, and everyone has their own definition of big data. So in our facility, it can come in a variety of different shapes and forms. We can have single large files from really long time lapses. So like this example here, where it's many thousand volumes captured, every second or 1 volume a second for thousands of volumes. Or it can be a mixture of multiple different files that are probably a little bit medium in size, but they collectively make up a big data set. So we can have tiled imaging of, high resolution pathology slides, and we can have, varying imaging methods where people take multiple time lapse in, high throughput well plates, or people just taking highly processed lattice volumes. But the kind of general consensus we have is that these data sets that they produce are just beyond the realm of their laptop or most workstation computers that we were using. So to go back to the pipeline that Jane's outlined, we can begin to look at the pinch points that are occurring and see how we can fix those bottlenecks from the acquisition through to essentially publication. On the acquisition front, we felt that we had at a pretty healthy level. We had those NVMe RAID cards, fast network, and the managed storage system with RDM here at UQ. On the visualization analysis, we could direct our users to CVL desktops or our own Windows-based VMs, and they can be connected then up with commercial or open source software for their analysis. That processing step in the middle is where we could really see some improvement. We weren't really getting effective use of the hardware or the licenses that we had for the commercial platforms. It wasn't easy for our users to access the HPC systems. So just to show you some of the data, the acquisition PCs have that really high-speed RAID cards where we see about 12 gigabytes a second read-write, and we see about 2 gigabytes per second transfer on some of our data migrations. The data storage is managed really well with the RDM platform, where we can isolate the data sets down into their individual projects. So people aren't kind of looking through for all those different data sets, and it's all organized, and we can have our collaborations and that on there as well. It's got the nice dashboard that manages and helps the users and ourselves kind of visualize what's happening. For the visualization analysis, we've gone away from those Mac Pro PCs to instead doing everything remotely now. The pros for the remote computing are that we can provide an experience that's way better than the end user's laptop. And we can keep the data secure in the data center and not out on the portable device. Upgrading the components of the servers is obviously easier and a better return on investment than keeping multiple either workstation computers or the end user's laptops up to speed to deal with this stuff. It also means that you can access the data or software from anywhere as long as you have an average or better internet connection. So here we have a bit of a game with WeHi, the stupidest places to do image analysis. And you can see that you can do it from your mobile phone if needed. But there are still challenges with this type of model and delivery. A lot of the software either just doesn't work for remote computing, it's license locked to avoid that, or it's specific for certain OS systems. And you still need that fast underlying storage layer to present the files to that remote session at speed. Otherwise, you've got a really powerful computer that's hamstrung by slow load times. Then there's also user bad habits. So they can be opening software and chewing up licenses when they don't need them, or not closing down or logging out of the system to end the session. And we've seen some weird quirks with different OS issues with Server or Windows Professional. So the remote desktops are really good, but they do present as a kind of finite resources that don't work with the even larger workflows that we're seeing with these new modern systems. So we wanted to create a tool that would speed up the processing, and we knew it would be on HPC. So we had to begin to kind of plan and flesh it out as to what would be required. We knew that the HPC environment would give access to way better hardware than we had, with faster IO than our remote servers. And we knew that some of our commercial software for deconvolution was optimized to run just stupidly fast on multi-GPUs compared to existing either interactive jobs or the older CPU horrible program that we used to use. And we had the goal of being able to string together multiple tasks, because we thought it would be great for the user to be able to just say, here's file, do this, this, and this, and then save it and back it all up for me. So a survey from the Broad Institute in 2020, looking at what the community have and what they want for future bio kind of image tools. The biggest #1 keyword that came out was that it should be GUI, user interactive, and user friendly. But if you look at the other side of the equation, which is what the bioimage analysts want, it was that they're jumping up and down about reproducibility of the data processing and ensuring that all the right parameters are being recorded so that the data could be reproduced. So our tool needed to be easy to use, have better than what's available, because you're not going to convince a researcher to learn something new if they don't see a benefit. And it has to be flexible because their use cases change, file types change, data shapes change. And it has to meet all the kind of FAIR requirements, journal, rules and publication requirements and UQ policies even. So that all had to be taken into account. So I'm going to quickly go through or introduce the two topics that are important for the portal, or one of the two things that we're doing on the portal. So the Lattice was obviously a big driver in creating this tool. So the de-skewing is a transformation that's required for that oblique nature that it's captured at. So if we look at the cell and kind of represented as the fried egg example on the left, the slices that make up the volume are the rectangles at the angle. Now, if you put that into Fiji or conventional software, it's not going to open it as the trapezoid offset data set that it is. It's just going to put it all together into a square, and it's going to look really janky, and it's not going to work, and it's really useless. So we have to perform this step called de-skewing. And essentially what that does is it pads out the data set with more pixels so that you have an aligned data set. And this results in a bigger file that's about 30% bigger, and all the dimensions are slightly changed because of the way the angles shift things. But this intermediary file that's created is then what you're going to have to use for the downstream steps, so such as deconvolution. Now, deconvolution is a pixel reassignment method that results in increased signal to noise in the output images. So hopefully these before and after images help you appreciate the power of this method. And what it actually really is a benefit for is the downstream applications like object detection and segmentation, and also just visual aesthetics that make it something that slots in that processing step for the visualization analysis. So for the people who don't know what deconvolution is, just really quickly, if you imagine a single point of light that's smaller than what we could resolve, it in theory should only take up 1 by 1 pixel, but light spreads out. And so we can measure how it spreads in that behavior as a thing called a point spread function. And that's just how the light is scattering in our optical system. We can then use that model to correct the raw image for all the light scattering by reassigning the intensities on a pixel by pixel basis. This correction can be really sped up with GPU parallelization, and obviously has a big benefit when you have lots of pixels, like when you've got lots of volumes in a Lattice data set. So as James kind of mentioned, we now had powerful computers that we had to get data to the IMB cache for the data store. That would then be moved to the QBI cache through the RDM layer. And then we would present that data up into the HPC and then onto the desktops. So that was our kind of workflow, but we wanted to make sure that it was simple for our users who are point and click, button push Windows classic users. I was one of them. So firstly, the data migration from the PC to the local RDM can be made easier for our users by auto-mounting the collections that they have read-write access to off that IMB cache. So they don't have to remember the file paths, or they don't, connect via a different route than what's been optimized for this building. Pitchy is a tool that was developed by Hoang that automatically manages and transfers data from the acquisition PC to the RDM with additional metadata tools for processing. And that was presented in the knowledge share last year. So if you want to learn more about that, highly recommend going back and watching that. The RDM layer handles the movement of storage from one cache to the other, meaning that it's going to move the data from the close acquisition PC computer. Now it's going to be available close to the HPC, and it keeps it all synchronized. But that data layer could also be managed and moved by other tools like Globus or RNet or SFTP. Now, the image processing portal that we're 20 minutes in, and it's the first time I'm really mentioning it, is what connects that last part of the pipeline. It's the simplified access to the HPC job submission for visualization and processing. So what it is it's a HTML website that has a GUI for simplified access to HPC image processing. And the reason we chose to put it on the web is that we don't rely on the end user to update things. We can drive all of the change from our end. It's fast and familiar with file browsing and processing menus that are intuitive, and it generates and submits the jobs to the HPC on behalf of the user. So there's no Slurm scripts or anything required there. And it's fully integrated with the CVL virtual desktops here at Wiener. And it's designed and funded by the IMB microscopy internally and the UQ RCC computing. So at a really high level, the IPP can currently do lots of just common tasks. From the web interface, a user can perform file management, CVL desktop launching and integration, file conversion tasks, image processing tasks, deconvolution, and they can manage their jobs that are running on the cluster as their own each individual built-in modules, which I'll go over in the demo. So let me explain more there. Users can request access to use the portal via our landing page, and that comes to us. And off the bat, we have some kind of high-level quick wins that are basically, we have the wizard-like workflows that are easy to set up and intuitive that the users can really quickly use and get up to speed with. There's familiar software that's integrated at the end, so it's a CVL, so we don't just go, off you go and good luck. They can go off to CVL and use programs that they know. And the software licenses aren't required to set up jobs or submit them, so we have good utilization there. And we have, what I kind of call managed HPC usage, which means that users, i.e. someone like a biologist like me, can't accidentally run inappropriate jobs on the head node or something. And that all the data is processed within the IPP is fair and it's backed up, and we control all of that so that we're not accidentally leaving it on scratch or not doing good behaviors. The following slides, I'm going to skip over really quickly. They're more for the people who don't want to watch the Zoom and just want to get the PDF that will be uploaded after the talk to give a bit of context around the different tabs. But I'll cover these things in the demo. So we just have some screenshots of the different sections. Before I jump into the demo, I'm just going to cover one of the kind of topics that we can do, which is really the driving force behind this. So processing Lattice Light-sheet data sets from the output into de-skewing them and deconvolving them. So what the portal does is it does a lot of things in the background automatically, so the user doesn't have to think about it. So I've listed the automatic stuff on the right and the user kind of required steps on the left. And this is probably a very oversimplification of what's happening in the background because I'm just an idiot biologist. But for this scenario, we can link together the input, the deskew, deconvolution, output, and then copying back to the device, all in pretty minimal user required steps. So we're going to go through the demo of the system, and then I'll hand over to Hoang to do the kind of how it works in the background. So here's one I prepared earlier. The image processing portal landing page has a bunch of information about, how to log in, documentation, requesting access, contacting us as well. So I already have logged in. So once you do log in, I have to have MFA and all those things as well. So I've just done that already for the demo, but we land on the landing page and we have our modules down the side. And this can obviously grow and expand as different required tools are put in. So the first one I'll show is the file manager. So what this is this is in the browser. We can navigate the file system that's attached to the HPC, and you can see that we have all these different collections, and that can be quite overwhelming. So we also give users access to the collections that they have read-write access to. Now, for most users, this will probably only be one or two in their local Scratch drives. I obviously have a lot of projects, so I've got a fair few here. And we can also bookmark any folder layer so that you can quickly jump to them. So I've got an RDM for doing demonstrations, and it's this one here. So this is the collection. And the files today, as much as I talk about big data, I am using ones that are slightly smaller than usual, or only subsets of the data sets, just for the interest of time. But you can see that as I move around to the files, I've got these folders, and I have my breadcrumb trail that I can move up and down through the layers, which is familiar, because most OS systems or Windows systems at least do that. I can also do some kind of file filtering. So if I search for only CZI, I would have only the CZI version of that file. Likewise, I can have TIFF files and I can search for those. What I can do is I can also create folders on the HPC. So I can create a demo knowledge share folder, and I can then take this file and I can, copy it. So let's put that folder into there. I can either move it, so in other words, make a copy and then delete the original, or I can just make a copy, so I can do that. Now, this is what I think is a really useful job because it's doing it on the HPC. Now, a lot of our users are going to take their big data sets and try to copy and paste them because they're wet lab biologists, they do Command C, Command V, and they don't realize that they're pulling all the data from one location via their laptop and then back out to the other location. And if these are big data sets, then they're probably going to close their laptop, unplug it from the network and go home and then complain the next day that it didn't work. So this is all doing it on the HPC side, not on the, via the user's laptop. And it's submitted as a job. I have a CVL running, and that job's running on the HPC to move that file. Coming back to here, I can also launch the files into the CVL environment. So I have already spun up a CVL environment, but what I can do is I can select the file, come up here to our desktop launcher, and I can see that this job's running. It's been running for an hour, and I can select different programs, and I can open the file, the program, and it's going to copy it from the Scratch location, from the file system down to the Scratch on CVL, and then open the program. So once I go to my CVL desktop, you can see it's copying it, and it's going to open Fiji, and then my data set's there to go. So if I'm a busy biologist, I can tell the portal to open my big file that might take half an hour to open, go away, do something else, come back, and it will be all open, ready for me to then interact with it. So we can see that we've opened, this is only a 20 gig file, but we've got our full Z stack and our time series type data. So that's quite nice. I can also, probably to the fear of some people, I can delete files. So that thing that I've copied, that job's finished. So I just check on the job page, don't need to do an SQ or anything. It's there, it tells me that the file's been copied. I'm going to come over here and put it in the trash can. Now it warns me and it's gone. So we can manage data in the cloud, not in the cloud, in the browser, in an intuitive way that's really nice. We can rename and do other tasks as well, but I don't think for this audience I need to show you. Now, I'm going to kill the CVL session. I'm going to bin that job, and I'll show you here. We give 3 options to spin up the different CVL flavors. So we've got classic, moderate, and maximum. where we just give different sizes and the users can do that. They don't need to log into Strudel on separate pages. It's all here in this page. Now for the file converter, one of the problems we had was one of the microscopes that we bought came with a file format that didn't work with bioformats. It just would not open. So we had to write a converter to convert it from the broken IMS file into just TIFF volumes. So what we can do is we can come in to select, sorry, I'll show you this. This is the file input module, and this is the same for all of the different kind of modules. We can add single files, we can add folders or series of files. So if I've got lots of Z stacks that are all the same shape and I just want to process them, I can add them this way. I can delete individually selected or all of them. So I'm going to launch our file browser, which is, looks the same. I'm going to go back up to the parent level of the collection and come into the Dragonfly dataset. So I've got a pretty tiny 2.8 gig file, but for the purpose of the demo, it's going to be sufficient. And I can select that. And I'm going to call it just KS Thursday. So that's the Knowledge Share Thursday output. So it already was making an output file called converter output within the input location. And I can then submit that job, and it's going to go off and do that. So we get a little green down in the corner, and that's going to be running over. And check on the jobs thing and see that, yes, I've got my CVL job running, but this new job is now submitted and is in the queue to run. The pre-processing tab, I'm not going to go through every single page because I think it's a bit of a moot point to this audience, but suffice to say, we can do different things, not just deconvolution or converting files. So perhaps we want to deskew the data by itself, or we want to combine files or average files. We can do that here. Did Roland have a question?
Yeah, if it's okay. My name's Roland, I'm from WeHi, and so I've had some chats with Niall over at WeHi around this.
Yep.
And one of the things that he mentioned to me, I just wanted to see if it was similar in your workflows as well, that when you're doing the actual capturing of the videos on the instrument, you're saying it'd be great to know as early as possible whether those videos are worthwhile keeping or not. And he was asking about doing the deskewing, and in his words, I'm sort of parroting him, I don't know what it really means. He was talking about the deskewing and also creating a MIPS file to be able to double check that that was his first quality control pass. And if it passed that particular one, then he'd be happy to continue doing the processing. I'm just sort of wondering, is this a similar situation that you're seeing in with your wet lab researchers? And if so, is there any automation, or is that part where Hoang's going to be talking about to be able to check that data quality before it gets moved across?
Yeah, so Niall at WeHi runs the Lattice Light-sheet that they have. They have a home-built system. We have the 3i commercial system, which is kind of pretty much the same instrument. And then there is now also the full commercial Zeiss white box instrument. The capturing the data on the Lattice, it's not like a normal microscope. There's no eyepieces, so it can be difficult to know if the data is kind of good. And if you are going to be generating a terabyte, you want to know that it is useful. So completely agree with that and Niall's sentiment. So with the home-built system, you have limitations in LabVIEW with how much you can see at the time of capture. On the 3i system, you can certainly see a little bit more, and you can dynamically go back and look at what's happening with changes in the data set. What we did to kind of overcome one of the issues that's kind of relevant to this is, instead of waiting to the end of the file to be processed and then ending up with a giant TIFF, slide book file in the case of 3i, we could chop it up on the fly, and then we could look at all those volumes individually. Now, I will say that our system's pretty robust, and we tend to not have to bin whole data sets. Where it can be useful is when you're capturing a very long time and you're looking for discrete events within that. So you can definitely create MIPS, and there are tools like Napari that are doing that kind of single frame processing. What this is, where this benefits is that it can do the HPC processing faster. So what used to take us, days or weeks to process, it can take seconds now. So I agree with what Niall's saying, where you may want to look at a single frame now, but I also think that you want to process it to actually understand the data, because going from that non-deconvolved to deconvolved really clears up, and you get that clarity in what's actually happening. So it's kind of like judging whether the data is good or not, where it's kind of foggy and not as sharp.
Yeah.
We do have another project with UQRCC called Phoebe that does non-sequential parallelized live processing of the data sets too. So we can convert on the fly from the slide book format to TIFF and then visualize them, but that's not part of this portal currently.
Yeah, there was one other small question that I never really got clarity on is when you're looking at the Zeiss white box version, the commercialized one.
Yeah.
I got the impression that at WeHi anyway, they were just relying on the Zeiss blue software to do some of the processing on that. But I wasn't sure if you could actually run open source tools and get the same results, or what's the, if you could talk about that, that'd be great.
Yeah, so the de-skewing is certainly not, only commercial. There's open source, and there's now, just the other day, open source GPU accelerated de-skewing. So that's that going from that kind of slanted back to a normal traditional shape. Built into the Zeiss system is another tool that lets you go from having it's another affine transformation that lets you go from scrolling through the data set kind of at the side view to a more classical top-down view for the users. It's generating another file layer that's personally not relevant if you can throw that straight into a 3D viewer that you don't need to see single slice info for, whether that's Napari or Fiji or commercial software. So Zen Blue is certainly good. It's a commercial software that requires licenses and money, and so the Zeiss system is a bit more restrained compared to the open breadboard type systems like the original and 3i.
And it only runs on a desktop PC. It doesn't run on HPC, Linux, or anything else.
But it's easy to be able to take the data from the Zeiss machine and start using open source equivalent tools. Has anyone done any testing on that in terms of whether they get similar results, or is it deterministic? Or
you can definitely take it to open source tools. The bottleneck that we see is bioformats. So if you're using or relying on bioformats to open a file, a terabyte file into Fiji, it's going to take you half an hour because it's not written for high speed opening. Whereas if you open that same terabyte file into the Zeiss platform, it opens instantly because it's written to do that. And it's those tools that aren't really open source and available for the high speed openers, which is why the route we've taken is to strip it down to another format, in this case TIFF, and then that can open very quickly because there are high-speed TIFF openers. Does that answer the question? Or?
Yeah, it does. Thanks very much.
No problem. So in the interest of time, I'll quickly just go on to the deconvolution, because this is a slightly different module where it's a more complex workflow. So what I'll do is we can add in, sorry, I don't want to add a single file first. Let's add in multiple files. And so if I come back up to my Lattice data sets, so in here, I've got basically just a very small snippet of data. So it's 50 time points from a 14,000 time point data set. But I can add these in. So I could, if I only had like C1 and C2 as a delimiter, I could put in, C1 and search for that, and it would only select the C1 for like my green channel if I want to process them separately. So that's how I can use the glob filtering. So I'm just going to select these 50 files and add them in. and it should read them in. I've done this correctly. Yes, so we've now added the files and I can work my way through. One of the kind of caveats to the Lattice system is that because we are going to tell it here what's the files, the image pixel sizes of a file that doesn't yet exist, because we haven't de-skewed it yet, I have to input the data. So I just know that it's going to be these numbers for this data set, but the user would know that. And then we work through these wizard-like steps. So we have to then, we're going to de-skew and deconvolve. So I need to present it with a PSF file. So I can tell it that the PSF file is located here. And I can point at that file. And that has metadata because that is all ready to go. And then we are going to de-skew the data. Interestingly, what causes some kind of potential issues with our users is we're not going to keep the de-skewed files. We don't need to. We can regenerate them at speed on the cluster. So we just keep the intermediary for the downstream step and then delete it. We can regenerate it with confidence. And I can just edit these parameters. So these are our camera pixel sizes and the voxel size. Now, again, these are kind of standard numbers. I can tell it how many iterations because deconvolution is cyclic, and I can then also work my way through. Yeah, Jay?
Hey, just a quick question, Nick.
Yep.
My understanding with the Lattice Light-sheet data is that normally you do the deconvolution. Sorry, you do the de-skewing and then the deconvolution.
Yes.
So is there anything in the portal to ensure that happens, or the user just says,
Yes.
no, they've got to do it that way?
Yeah, so what I didn't kind of point out is it's, for me, it's defaulting to light-sheet deconvolution. So if I was to change this to a different modality, then I wouldn't get the de-skew option.
Right, okay.
So even though we're kind of pointing to the PSF, which is the deconvolution aspect, the de-skew does happen beforehand.
Yep.
And it won't let you advance without completing each step.
Yeah,
Okay.
so
Okay.
as we move along, you can see that they're changing color. I don't need to do any of these filtering steps for this data set. I can choose to do decon. In the interest of time, I'll quickly submit the job and then we can have a question while we run it. So just quickly, the portal automatically looks at the data and suggests, how much memory is going to be required on the cluster so that we're not just asking for the maximum like a person may do. How many nodes to run that job on and how many GPUs to use. So we then get kind of a summary page and I can submit it. What's quite nice as well is we can save this workflow as a template because they're just variables that it's naming right into the spreadsheet, into the Slurm script or batch job. And so I can just call this demo as the template. And so if I save that, I could then come back up here and, work on a different set of data. Just a single file, load that Thursday template file. Work my way through. It's remembering all the settings for me.
Yep.
And then submit it. So if you want to ask the question.
Thanks, Nick.
That's right. Was there another person with their hand up? I saw the little Zoom pop up. If not,
Yeah,
basically, sorry, yep.
Yeah, it's just Roland again. I was wanting to ask, can you save that template so that you could run the job automatically straight off the instrument, so to speak, so that if I was to say to someone, hey, here's my sample, can you run it? And here are the parameters I actually want to run. Do you know that sort of thing beforehand, or do you have to, does it depend on the acquisition that you might change some of these parameters?
Or if you're relying on a biologist for, doing the same thing over and over, it could be dangerous. But yeah, more or less you can. Most of the settings are the same. With the Lattice, you can change lots of parameters. It's really capturing the PSF that's matched to those parameters, because that's going to be the biggest effect. Things like the D-skew angle is not going to change from session to session. The number of cycles, you can either define it, or depending on the deconvolution engine, you can say, okay, this is keep going until it has a cutoff that it's determined. So in the to answer your question, I guess yes. At the moment, in its current instance, it can't do that, but that could be something.
One of our ideas is to use Pitchy to access those pre-saved templates. So when you're ingesting the data straight from acquisition, you could set up a job and it'll just go once the data's there.
Cool, thanks. That's very handy to know.
So just, I'll quickly show, we've got our jobs running or submitted, sitting in the queue. We have an admin tab that lets us kind of see all of our jobs and we can search them and things like that. And we have documentation for our users on how to use it. And obviously this stuff is on GitHub as well. Lastly, I'll just show that I have, when we submit the jobs, when they are finished, the user gets an email and it says, you know, your job's done, your files are here. So this was the TIFF converter file. It launches straight into the portal. And then I could say, okay, take that file that's now a TIFF stack and let's, you know, push that to our desktop. And I can come back to, I don't have it, so I can show the desktop. And then it launches that CVL session again. It's opening PG, copying the file, etc. So it's kind of like a full loop. Submit the job, you get the email back saying, okay, your job's done now, you can launch it, and off you go. So in the interest of time, because there are a few more slides, I'll jump back to the presentation and I'm going to hand over to Hoang to talk about how it actually works under the hood for the technical people.
Thank you, Nick, for the wonderful demo. Now everyone must be curious on how it works under the hood. Well, that was a joke, by the way. Normally people don't care about it. Anyway, so in the back end, the way the portal works is pretty much the same as pseudo web or CVL. That is the HPC, in this case, being just a certificate issued by the portal backend. And then the user actions from the web interface would trigger corresponding SSH commands executed by the portal backend in the Vina head node and under the user account. The results are then sent back to the front end and displayed to the users. Next, please, Nick. So when we first developed the prototype, in order to cut the development time, we actually reused the backend from Covestra. So for those who is not aware of what Covestra is, this is a virtual desktop running on the cloud, basically a modification of CVL. And for the first iteration, the user interface can be seen on the right side. We basically just copy the user interface from Microvolution, which is the deconvolution package that we are using. I guess at that time we're still learning anyway. But for the second release, we completely overhaul the user interface. And we've added quite a lot of features in terms of files management and the wizard-like settings for setting parameters that Nick just showed. And we managed to integrate CVL at Vina as well. Okay, so for the roadmap, so before this talk, we have already started working on the next release, which was expected to deliver around March. That adds a couple of new features. The most important one, probably the support of open source deconvolution engine. And Felipe, our intern at RCC, probably somewhere in the audience, have made quite good progress with PyCUDA Decon. So that would answer the question, Roland, of the support of the open source deconvolution engine. Secondly, I've been working on improve our metadata handling. So the idea is if the portal is aware of more metadata fields, we would implement a smarter UIs based on the file types that that's been loaded to the portal. And I will make some improvement on the admin page as well for user reporting. And another item is we are looking into supporting more converters into the portal because with facilities like IMB or any other microscopy facilities, the users normally deal with many different file types, and it is very tedious and time-consuming to convert between file types if it's done in in their workstation. If it's done in the HPC, that was speeding things up quite a lot. A little bit further, we would like to integrate more tools or pipelines into the portal. So for things like more tools like min-max projection or filtering, of more machine-specific tools like dual camera alignments, etc. We also consider improve the files management even further. So things like we would like to get more stats from related to files or collections, or we would like to integrate the files movement tools such as globals into the portal. So at the moment, the main users of the portal are from IMB and SPMS, but we are promoting it to more users within UQ. And we're looking for domestic partners likely when our open source Decon is available. And ultimately, we would like to have the ability where the users can divide the computations in terms of workflow and run from the portal. So for conclusion, microscopy data is getting bigger, and often the hardware access and ease of use for available tools are often the bottleneck when it comes to processing microscopy data. And our image processing portal is a collection of managed tools to lower the barrier of entry to HPC for a non-traditional user base. So that is good for users because they can scale up their computation beyond a single node. And it is good for admin as well because the users cannot submit inappropriate jobs to the scheduler. And in the future, we will look into integrate workflows into the computation so that it efficiently use resources and licenses, discard intermediate files, and provides provenance-rich metadata following the FAIR principles. And of course, we would like to grow this platform with other organizations contributing their tools into the portal for the benefit of the community. To get this portal up and running at this stage, it requires efforts of many people around UQ, but these are the main players. So with that, I would like to thank you all for listening and welcome any questions.
Thank you so much. Thank all of you. I just want to say a big, massive congratulations to you all for getting this up. I think it's absolutely gorgeous. It's brilliant. And, you know, Nick always speaks so well and explains things so clearly, as you all do. But I love that it's so clear that this is exactly what a biologist is going to want. So it's like all of the cut down best bits that they need from CVL and from the other things, but it's presented in a way where it'll be streamlined for them and they won't be distracted by perhaps some of the other tools or packages that are available. I want to ask a little question, if I may, to start. We've only got a couple of minutes, but in the beginning where you've got the file tree, some users might be concerned that other people would be able to see their data. Is that the same as when you would go and browse through CVL? You can see that it's there, but you can't access it or touch it in any way?
Yes, it's the same.
And the other thing I wanted to ask was, I saw there was a tab for electron microscopy. Are there any plans to do similar sorts of workflows for CMM or?
Roger?
Yes, for sure. We have a discussion for cryo. EM, but also on other software packages. Depends on the software package they can put up into CVL. PG has been born out of CMM activities. So I'm glad to see that others can benefit from the Clouder install here. And I think we were a community, we try just to push it. I can't tell you which software package will be the next one we will be working on, but yeah, we have segmentation as one of the highest priority for volume imaging and single particle processing for cryo-EM.
Sounds good. We'll ask another question too, which is with those parameters, Nick, that people were inserting into the workflows there, is that information that they would get from the machine, or do they get that from the literature, or how do they come by knowing what they're going to be putting in?
Yeah, the pixel size of the camera won't ever change. So that's basically a fixed one that could be locked down. The thing that's going to change is the Z spacing, and therefore when we deskew it, the after deskew, what the voxel size will be. So they generally don't change for our users, but for a few fringe cases, they will change so that we have that flexibility to add. It's something that probably could be predicted because we have an algorithm that calculates what one is from the other. So that's just more tools that could be automated in the back end, but hasn't been a major priority because no matter where you are, if you're doing lattice processing, they're numbers that you're going to deal with from that device. So
one of the biggest issues is even the commercial packages don't read all the metadata in, or they get it wrong. So we're spending a lot of time in the back end at the moment, hand going through typical data sets from all our instruments and correcting those mistakes so that we'll read it all in automatically and make it really easy for users.
So down the track, do you see that step disappearing, or?
Yeah, at least filling it out and giving like a green tick or whatever to say it trusts that, or this one's dubious and things like that.
Does anyone else want to ask any questions?
No one's got a question. I've got one.
Yes.
Yeah, I'm sort of curious, when you're talking about domestic partners, are you working on a model where they would come to Wiener to use this workflow, or they'd be able to take an IPP and put it onto their HPC of choice? And if that's the case, how easy would it be to replicate the glueware between the HPC part and the web part?
I can answer the first half. I'll leave the technical to the IT guys. Yeah, absolutely. Our model is that people would take this and run with it, just like CBL or anything else. So, you could install it at Monash, or we hire your own hardware. And certainly we'd be open, to begin with, people trying the data sets out using Globus to move it to Wiener to try it there, see if it suits their needs and help develop tools on it. But really, we're looking to make this a standard thing that everyone can use. And really, we're looking for partners who can contribute coding, because a lot of places have their own hardware, but developing the tools is the hard bit.
Yeah, and I'm sort of wondering, when you were talking about the open source tools that weren't necessarily available for some of the positions, if you'd looked at Google Summer of Code and Outreachy for getting students to look at that thing, and maybe doing it as a national kind of focus for, say, the newly formed Research Software Engineering Australia New Zealand community, because I feel that there's a lot of MRIs, medical research institutes, that need this kind of thing. And I think if we all work together in an open source kind of community, we could get a lot more done just chipping away at it from a lot of different areas.
Yeah, look, agreed. We're looking at all sorts of ideas. I guess our biggest priority is getting a tool up and running as quickly as possible to suit our needs right this instant. But we're sort of coming to the end of that process now, where we have something that does all of the major stuff we need. And now it's, well, where can we go with this and make it better? And so we're just in that process of transitioning, where we want to have these discussions with people and, go, what can we do? And we're having ideas about talking to Google and their 5% idea, where their staff can work on side projects. And it's really spitballing. How do we fund it? How do we get people working on it? How do we grow it?
To answer the question, how easy it is to move this to another another HPC. I say that it's quite easy to take as is. Like, well, obviously, if you don't, when the open source deconvolution package is available, and if you want to use the same package that that we chose, then I think it's quite straightforward. But obviously, if you need some customization, then it takes more time, really.
Yeah, Jay has asked there, Hoang, if there's a repository, open source repository available.
Oh, yes. So I put that GitHub, yeah, right. So whatever we're doing is just open source.
Cool. Okay. Links in there. I'll add it in to the notes afterwards. Well, thank you again so much. I'll call it into questions because a couple of people are heading off onto their next meeting, and just say thank you again so much for your time today, and thank you for those beautiful, clear explanations, and big congratulations, and thank you everyone for coming along. I'll put this up, so if you have some colleagues you might like to share this presentation with who couldn't make it, I will be sending around the link later on today or tomorrow so that they can access it. And I'd just like to say thank you and invite you to come along to our next session. I know that when I introduced it, I said this was number 11. It wasn't, it was lucky number 12. And our lucky number 13, next time we'll have a change of pace for our next knowledge share, and we'll be going across to WA, where Dean will be talking to us about the rollout of CVL on UWA. So he'll be bringing us up to speed with the advances that they're making there, and talking about how it's going to be deployed over there. And hopefully you'll be able to come and join with us again next month. Thanks, everyone. Thank you so much.
Thanks, Catherine, and thanks, everyone.
Thank you. Yeah.
Thank you.